blood vessels, you should be, I want you to be the blood. So trace the flow of blood. So you start with the superior inferior vena cava and you go to, be sure you're able to write down. You know in your head, you're picturing it. Oh, it will enter the right atrium. Then after that, AV valve, don't forget, right AV valve and right ventricle. Then you're going to say, this blood is low in oxygen or high in carbon dioxide, right? Then from here, going to go to from the right ventricles to the lungs, pulmonary artery. Then here to pick up oxygen and load carbon dioxide and go to the left. So be sure you know exactly, okay? Remember the pacemaker of the heart. So remember I told you that the origin is from the SA node, then SA node to the AV node. So in other words, if the SA node fails to fire, then the next one takes over, right?
Yeah.
It just takes over. That's their job. It's like if the dad is deployed, the mother takes over. Then if the mother is somewhere, the eldest, so they just take turns, okay? And then EKG, you should know EKG. Normal EKG, you should be able to estimate the what do you call this, the heart rate. Remember our magic number, 300, 150, 175, 60. This is just a guesstimate. And that will be good for you to remember when you start working. And then what else? Did I turn on? Yes. Of course, the cardiac cycle. You will be given a picture of the cardiac cycle, and I hope you remember. Just be sure you know where is ventricular systole, diastole, where in the cardiac cycle is there isovolumetric contraction, isovolumetric relaxation, the EKG tracings, the first heart sound, second heart sound, then your inotropic, chronotropic, you should remember that, right? So the following, let's say if you're losing blood, will that produce an inotropic or a chronotropic effect? And then will it be positive or negative? Okay? Then for the heart, remember that the cardiac output, remember the formula, you may be asked to compute cardiac output equals heart rate times stroke volume. And there's factors that affect that cardiac output. You have the preload, remember pre is before the heart, afterload is after the heart, and then the heart itself. So the preload meaning the load, maybe there's an overload. So give me some examples of preload, maybe too much volume coming in, right? Then cardiac contractility, maybe you have heart failure. Then afterload, so there's the artery. So preload is the venous return, afterload is the artery. There may be some resistance there may be some hardening, hypertension, okay? So be sure you remember the formula. Cardiac output equals heart rate times blood volume, stroke volume. And then for the blood vessels, you should know the difference between, you should know this one right here, right? Differentiate between the artery, capillary, and vein. And you know that the arteries and the veins have three layers. The tunica media, if you don't remember media, the inner, the middle, and the outermost layer. So tunica intima, inner, media, middle, external, the outer layer. So let's practice. If a patient has arteriosclerosis or hardening due to cholesterol plaque, where will the problem be here? What do you think? Tunica intima, media, or externa? Intima, right? Because the cholesterol plaque is there. Very good. Okay? So just remember to answer you, because you will be asked. Let's say there is beta blockers, which is I hope you know what beta blockers do. What do beta blockers do? They are muscle relaxants, right? That's why we give. You know that people with anaphylactic or asthma, they're given beta blockers to relax the muscles. So where will the beta blockers do their job? Intima, media, or externa? Media, because that's where the muscle is. Muscle relaxant, okay? You know, you understand the word resistance. So resistance is the one that is that force that the blood vessel has to overcome, or even the heart has to overcome, so that it has to pump stronger to overcome the resistance. So that, what do you think? If there is, we're just talking about the blood vessels, if there is any constriction, like there's a plaque right here, right? The reason why we become hypertensive is because there's increased resistance if there's a plaque. Meaning, when you decrease the opening, the diameter of the opening, you are increasing the resistance. Does it make sense? So, I hope you know what resistance means, okay? Anything that, if you don't remember, you ask me while you're taking the test. So, this test, you will take it here, and hopefully you take advantage of what the pointers that I gave you. Okay. Then you're also going to be asked for to compute for filtration, net filtration. So we're now looking at the capillaries. We studied our arteries, we studied our veins, then we're going to study our capillaries. Don't forget, for the arteries, another difference between arteries and veins, always remember the elastic fibers here, okay? Remember the elasticity right here, this picture. See those elastic fibers? That is very important, makes it ideal for the artery to accommodate all of that blood coming from the heart, then go back and pump all that blood. The vein, the vein is referred to as a reservoir because it has a very thin muscle layer. The blood just sits there. So it's up to our skeletal muscles to squeeze that. That's why between vein and artery, thrombosis or clotting happens in the vein, not in the artery. Arteries are very active, always pumping and squeezing the blood, constricting.
So I've had some patients who they go to stand up and they feel kind of lightheaded, and some are physical therapists have encouraged them to, like, when they have their feet in the floor, to kind of push against the floor. Are they trying to get them to just contract the
Yes,
muscles?
yes. Doesn't it make sense now? You saw how the veins are squeezed in between the muscles. Even when you're just pushing against the floor, your skeletal muscle is squeezing, is contracting. So that's good to squeeze your, where is that? To squeeze your, right here. See, it will squeeze the blood vessels. Where's mine? Right here. Okay. Even though your feet, see, it's just flat on the floor. This, your, even like here, you try to tense your mouth, you see the masseter contracting. That's the same thing that happens in your legs. The last part of this chapter talks about tissue perfusion. So we're talking about our capillaries now, the capillaries. Of the three types of blood vessels, capillaries are your exchange vessels. They have pores, they have openings. They even classify capillaries whether according to, depending on the type of opening that they have. You do not need to worry about this classification of capillaries. I'm not going to ask you, do you think this is a continuous, a sinusoid, capillary, or fenestrated? But this is just showing you that capillaries have openings, and depending on where you find this in the liver, which is the part of the body that grabs your amino acids and makes all your proteins for you, and proteins can be big, they have this kind of capillary, see? So that after they have made the protein, the big proteins can just escape and go back to your circulation, like your albumin, your globulin, fibrinogen. This is a common type of capillary you find in your body tissues. This one, you can find it in your endocrine organs, which makes the hormones, and then they secrete it into the circulation. See, but whether it's this or this or this, what you need to remember is capillaries have openings to allow entry and exit of substances. So anytime you're asked, what blood vessel do you think will white blood cells leave the circulation? Here. How about you took your hormones? How do they go to the target organs? Only through capillaries. How about germs, bacteria? How do they enter? Through capillaries. Unless there's a break, then bacteria can enter. But when it's time for them to spread to your body tissues, they can only do so through your capillaries. So the main job, knowing that this is the third type of blood vessel, we should know then, remember that the job of the cardiovascular system really is what you call perfusion. Perfusion. I think you know what this means, right? Perfusion is to uh, supply fluid to your body tissues. Where did the fluid come from? It was in your blood. Then when that fluid escapes your blood, that's what you call tissue perfusion. And tissue perfusion happens because of different forces. So here, this is how perfusion happens. It can pass through diffusion. You know diffusion, we're moving from an air of high concentration to low concentration. That can happen endocytosis, exocytosis. But the most predominant way for perfusion to happen is called bulk flow. And it involves pressure. Involves pressure. So you will be asked in your lecture test on Wednesday to compute for filtration pressure, net filtration pressure. So I'm going to draw right here again. There's your heart, right? There's your artery. Then there's the capillary. Then vein back to the heart. This is the normal direction or the most predominant way for blood flow in your cardiovascular. The blood flows from the heart, then it goes to the capillary. From the capillary, it branches to, artery. From the artery, it branches to capillaries, and then capillaries, vein back to the heart. You know that substances should escape or leave your capillaries. That's the purpose of circulation. You want your oxygen to leave, to go to your tissues. You want your vitamins to go to your tissues, your proteins. But how do they move? How do they leave your circulation? They don't have legs. They can't swim. They are pushed out. It's the pressure that will push them out. So there are two pressures here, right? The pressure pushing the substances in the capillary out, and the pressure that is keeping them back inside. It's called osmotic pressure, pulling them back inside. Pushing them back out is called hydrostatic pressure. Okay? So two words, hydrostatic, like the waterfalls falling down, leaving, and then osmotic pull. So we have two compartments here. The blood compartment, which is in your capillary, the capillary compartment, and then this one here, interstitial fluid. So blood and interstitial, or you can say intravascular. You understand that intravascular and interstitial. So when I say blood hydrostatic fluid, this is pushing the fluid out of the blood vessel. When I say interstitial fluid, hydrostatic pressure, that means from here pushing there. So hydrostatic, wherever it originates, it's pushing it out. Osmotic is sucking it back in. You will be asked to compute for your test on Wednesday. So you will be given pressures and you will be asked, compute for the net filtration pressure. And then I will ask you, given that value, where do you think, what will be the direction of blood flow? Into the, out of the artery, or is, my question will always be, is this normal? Where will this value be normal? In the arterial end of the capillary or the venous end of the capillary? So this is not hard, okay? This should be a freebie. There's two pressures, hydrostatic pressure and osmotic pressure. When I say blood hydrostatic pressure means from here going out. It's, in other words, that's your blood pressure pushing substances out. But the opposite, so you see, interstitial fluid, hydrostatic. So the interstitial fluid may also exert some hydrostatic pressure that will resist that. So it's going to push inside. Here in this picture, the interstitial fluid hydrostatic pressure is 0. Then there's the osmotic pressure, the blood osmotic pressure, this one right here. They include this colloid oncotic pressure. It's because it's the proteins that pulls it. The osmotic pressure, right? Water is attracted to where it's more, there's more concentration here. The proteins here pull it. So the blood osmotic pressure pulls the fluid back here. And then interstitial fluid osmotic pressure, see? Meaning the proteins out here in the interstitial fluid will pull the fluid out there. You see that? So one more time, hydrostatic pressure, osmotic pressure. Hydrostatic pressure pushing it out wherever it's originating, and osmotic pressure sucking it back in. You understand the two pressures? So you need to remember. Use arrows to help you remember. Okay? This one will come out in your test for sure. And you will be asked to compute. So let's see how they computed the net filtration pressure. This is the capillary, and see the blood flow. This is from the heart. This is the artery end. A capillary has two ends, arterial end and the venous end, right? So heart, artery, capillary, vein back to the heart. So there's two ends. One end of the capillary is the artery end, and the other end is the venous end. So here, the red is the artery end, and the blue is the venous end. So here, look at the hydrostatic pressure. Let's compute the hydrostatic pressure at the arterial end. The hydrostatic pressure is 35. So what are the forces pushing out? There's an interstitial fluid osmotic pressure. What does this mean? What does interstitial fluid osmotic pressure mean? The proteins here, right, in the interstitial fluid attracting the fluid here. So these two forces are pushing the fluid out. You see that? So the forces out, 35 plus 1, minus the opposing forces, which you have the interstitial fluid hydrostatic pressure, which is pushing it there, that's 0. But there's a pull from the proteins here. So there's proteins here, and there's proteins here also. The proteins in your blood vessel, you remember? Albumin, globulin, fibrinogen. So especially albumin. Isn't it the job of albumin to maintain blood pressure? So this albumin is sucking the fluid back there. It's opposing the hydrostatic pressure. So you see, 35 plus 1, that's the forces pushing the fluid out minus the pull of the albumin. The pull of the albumin is called blood osmotic pressure. Colloid is for protein, so it's pulling it 26 plus, what's this zero? This interstitial fluid hydrostatic pressure, maybe there's pressure pushing it there. So the net result is 10. This is a positive number. So a positive pressure is normal in the arterial end of capillaries. One rule that you should remember is this one, okay? So you remember your formed elements, RBCs, white blood cells, platelets, albumin, globulin, fibrinogen. The general rule is these 6 entities, they're not that big to leave your capillary. They keep passing through so that by the time you get to the venous end, there's the, right? It's not normal to find red blood cells in your PP or anywhere else. They're only normal to be found inside your blood vessels. You understand? Your white blood cells, yes, they leave when there's an infection. But if there's no infection, they should stay here. So they keep passing through. And so by the time you get to the venous end, you still see them, the albumin, the globulin, fibrinogen. And it's very important because this albumin is the key. You notice here at the arterial end, you needed a positive pressure to push all your substances, your vitamins, your minerals, your nutrients. But the main ingredient that you pushed out of your cardiovascular is water, which is the key for maintaining blood pressure. So all of that water that was pushed out, you need to bring it back in. What you needed to leave is just your oxygen, your nutrients, your hormones, but the water has to go back in. Do you follow? One more time. To push out your nutrients, your oxygen, your vitamins, your minerals, they are pushed out through being dissolved in water. So water was pushed out, and along with water was mixed in there your vitamins, your nutrients, everything. You want to leave your vitamins, your nutrients here, but you want to bring back the water because your cardiovascular system is a closed circuit. That's why that rule that these big proteins should never leave your circulation, that rule should always be followed by your cardiovascular system. So you see, because these big substances didn't leave, when you get to the venous part, this albumin is the key to suck back the water, bring it back into your cardiovascular. You achieved your goal of nourishing your systemic, your muscles, all your tissues, your skin, everything here. But your water has to go back in there. Do you understand? That's why people with ascites or you understand ascites is fluid in your belly. These proteins, these proteins are made by your liver. So when you eat your steak or your protein, you digest them into amino acids. Then those amino acids will, the first stop for all your nutrients is your liver. The liver is going to then start making these 3 albumin, globulin, fibrinogen. And so when they leave the liver and go to the heart pumps it, the heart then pumps it as this, and this. So it makes sense then. Did you hear me? I said the liver is the one that makes that. So if you have liver failure, like alcoholics, you notice with alcoholics, they have protruding bellies, they have very thin legs, there's muscle wasting, and then you feel you tap that belly, it's fluid in there. That's because they're lacking in this one. So they don't have any proteins to bring back the fluid there. So they have blood pressure. The pressure will push. They're able to nourish their tissues, but there's not enough pull to bring back the fluid there. And so you tap, they have ascites or edema right here in the abdominal pelvic cavity. It's called ascites. So let's look at the pressure here. You will be asked to compute. I know that. I'm looking at your test. So be sure you know how to compute, okay, for net filtration pressure. So let's look at this part. And after you compute, if it's a positive number, you know that this value is normal in the arterial end. In the arterial end, I want to push out. In the venous end, I want to bring it back in. So look at in the venous end, the number, the value is negative. So don't forget, you will be asked to compute, and then I will ask you, this value is normal in the arterial end or the venous end. So you will circle one choice. Let's look at by the time the fluid reached here, right here. Remember, this keep passing through, they don't leave your circulation. If, especially the RBC, if they leave, there's a damage. These capillaries are not, the pores and the openings are not that big to allow this to leave. But the white blood cell, you remember, they have that gift. They're able to squeeze out only this one. Okay? So look at in the venous end. By the time my nutrients leave, look at, compare the blood hydrostatic pressure here in the arterial end. It makes sense that by the time you get here, the blood hydrostatic pressure went down, right? Because you used that pressure to push out. So it's down 16. What else is the force pushing the fluid out? This interstitial fluid, osmotic pressure, sucking it out. So 16 plus 1 minus the blood colloid pressure. This attraction of the albumin, right? You see, the proteins, the albumin, globulin, they never change. They stay here. So the value is the same. It's sucking it back in. Plus 0, which is this interstitial fluid hydrostatic pressure. So here at the venous end, you have a negative value. Negative value means you're now able to um, uh, suck, what's that better? Attract the fluid back in here so that you maintain your blood pressure. The next chapter talks about the lymphatic system, which I want to touch on a little bit right now. You see this part right here? This is called your lymphatic vessels. Because here in this picture, we're just looking at a still shot. But what you need to know is the bus keeps moving, right? Your blood keeps flowing. There's no stop. They don't wait. Like the bus, right? There's a schedule. You have to look at the schedule. And the bus won't wait for you. If it says the bus comes at 6:15, you come at 6:17, it's too late. You wait for the next bus. Does it make sense? That's the same thing here. Okay? So this is just a still shot. We're looking, but the blood keeps flowing. So they should be fast. The water right here should be fast to follow this. If it missed that, can you imagine if it missed the first round trip? Then that means it can't wait. It's not like if we miss the bus, we can wait for the 6:30 bus. You can't wait. Why? If the fluid, if the water missed this bus, it can't wait for the next round trip because the blood pressure will be compromised. Okay? Which means that the slow pokes, and there are always slow pokes. The water is slow sometimes to follow that or be attracted to that albumin. But It cannot wait for the next stroke volume. So to solve that problem, otherwise you're going to have edema. To solve that problem, you have the lymphatic system. So the lymphatic system, see excess interstitial fluid. The ones that missed that first trip will join that first trip. It won't wait for the second trip. Whatever trip they were on, it will eventually join. So, like in this picture, this is my heart, artery, capillary, vein back to the heart. So, if this is the first trip, so it's going to go back first trip. It can't wait for the second trip. So if it was slow and the first trip is already moving, are you following? Even though I'm using the word first trip, okay? If it's as it moves, look at it's the blood pressure is going to be affected. Stroke volume for the second trip will be affected. We want to maintain that. That's why you have that lymphatic system. See, it's another vessel. It's called lymphatic vessels. And I'm going to draw it like this. And where it will empty, see, it will join like a shortcut. If police are chasing the bad cops, right? They go their own secret pathway. And see, the slow pokes will then jump here, the first trip that missed the actual main route, see? And they'll be quick. Return, they join. The blood pressure will not be compromised. Okay? So one more time, it's always the case. We always have slow pokes that will miss the bus. You cannot wait for the second heartbeat. You cannot. Because if we wait, then that means that the second one, the volume is down. Your pressure goes down. You cannot. That's not going to work. You're going to end up with edema. So that's the function of the lymphatic system. Okay? So, and that will be our next chapter. Do you have any questions on this one? Do you think you'll be able to compute for the blood flow? Okay? And that's the last part of this chapter on the blood vessels. Before we move on with this one, you know that there are several factors that can affect your blood flow or the flow of blood or tissue perfusion. Tissue perfusion is always the goal. Okay? So you have baroreceptors, you have chemoreceptors, you know the function of this epinephrine, norepinephrine, you know antidiuretic hormone. You notice that most of the hormones we're talking about, they have something to do with the ultimate effect is it always makes sure that there is a blood pressure. It maintains normal blood pressure. Otherwise, you're going to go into shock. Have you heard this word shock? Shock means there's no more blood flow. So there's shock because of low blood volume, hypovolemic shock. Vascular shock, meaning maybe there's severe anaphylaxis, severe allergy. That's why you can die from overwhelming allergy. So some people wear that bracelet to make sure that everyone is aware of their allergies, because you can suffer from vascular shock. Or if your heart just gives, then you go into cardiogenic shock, and this is the one that we do the defibrillator. Okay? Okay. So here's the normal flow of blood. So you see, look at the arteries. It's always a capillary bed, then vein back to the heart. So the normal flow, heart, artery, capillary, vein, back to the heart. This is also artery, capillary, vein, back to the heart. This one here is your liver. This type of blood flow right here is called portal circulation. So this is called a portal vein. This is my liver. This is the hepatic portal vein. This is what's referred to as hepatic portal circulation. This is very important. We're going to study this again when we take up our digestive system. But now, I want you to look at this one. What's a portal vein? A portal vein is a vein that connects two capillary beds. The general blood flow, I said, it should be artery, then capillary, then the vein should go back to the heart. But look at this one. There's an artery, then there's your, this is your GI. So there's your stomach. This is your small intestines. You know, from the stomach, you can absorb nutrients. Same with the small intestines. So these are your capillaries. What are the capillaries picking up from your small intestines? It's picking up my glucose that I just digested. Do you follow? My amino acids. Don't you agree? It may also be picking up some germs, like maybe the 10-second rule, or you eat, or even poison, right? Like they always come up with new findings. Like if we cook with Teflon now, they say recent studies show that Teflon has some chemicals and that can be mixed with the food that you, even the coffee. They say the coffee mug, and when you put your coffee in the microwave, some people want it hotter, put it for 25 seconds. They say take out that coffee mug. If you feel the mug hot, the mug itself hot, that means there's chemicals in the coffee mug that has mixed with your coffee and you're drinking those chemicals, and they can be toxic. Because there's so many findings, which means that it's possible that along with the nutrients, we are also absorbing toxic chemicals. Maybe what do they say? The food preservatives, like the food coloring, the food additives, they say that can also be toxic. We are absorbing that along with the nutrients that we just digested from our food, okay? So that's why this hepatic portal vein is going to be very important for you to appreciate. I have a liver worksheet for you when we get to the GI part. You need to appreciate what your liver does. So here, I'm also absorbing my amino acids, my fatty acids. See, look at, what's the first stop of everything that you absorb? The liver. So this is the vein. You think it will go straight to the heart, but it's making a stop in the liver. Your liver is so hardworking. What the liver does, I always picture this as little elves. You know, the elves make gifts for Santa, to help Santa. And that's what it's doing, right? Do you remember? What does the liver do? It stores glycogen, right? So it's grabbing the glucose that you just absorbed. It's packing the glycogen here to tide you over. Can you picture that? Then it's also grabbing the amino acids to make your proteins. See? So once it leaves, you already have formed albumin, globulin, fibrinogen. See? It's the liver that made that. So that's why people with liver failure have problems with making this protein, or they can go into protein overload. You limit the amount of protein in their diet. You remember, we're going to talk about that when we study our renal clearance. How of the different nutrients, protein is the most toxic if you have liver problems. And also your liver has phagocytes. So any kind of maybe germs because of the 10-second rule or any kind of toxic substances, that's what the liver does. Do you remember your smooth endoplasmic reticulum? Remember the job of that is for detox. So if we drink our alcohol, see, look at what the liver does. It doesn't even permit that alcohol if it can help it. It prevents the alcohol from spilling here as still alcohol. What the liver does is convert that alcohol immediately to fat. So when it leaves, it's fat that's circulating. And then your heart will pump it, and then your belly will store it as adipose. It didn't circulate as alcohol. But if we drink over the 0.08 alcohol limit, then the liver gets overloaded, it spills as alcohol, and so we feel the buzz. That's what happens. Do you understand this portal circulation? You're going to be asked about this again when we study the digestive system. Now you're just being introduced. This is what you mean by portal circulation. There is a modified type of portal circulation when we study our renal system. Okay? So, look at this one. Look at this picture. Okay? We're going to use this picture to understand why people with thrombosis will generally have, if you have thrombosis or blood clots, you always worry about the lungs. Why will the clot settle in the lungs and block your, what is it? What's the function of the lungs? Do what? Correct. Yes, the lungs is your way for oxygen. Is there any other way for oxygen to enter your body? There's no other way, right? You inhale through your nose, but eventually it lands in your lungs. And your lungs has the blood vessels to pick up that oxygen. That's how your oxygen enters your body. There's no other way. Not through your GI, only your lungs. So, can you use your book? Start with your femoral vein. So look at your blood vessels and tell me what, so go to your veins, because clots generally happen into the veins. And let's trace the blood flow. So right here. So you generally will have a clot, let's say, in your leg. So you see on page 743, table 19.9. Let's start with, let's say a clot happened in your tibial vein or saphenous vein. And then can you trace? Let's trace. What's the next vein? So let's go to the saphenous vein. Then next, going up, because it's supposed to go up to the heart, right? From the saphenous vein, what's the next vein? Popliteal. Okay, next. Remember, veins are big, right? Okay, next. Okay, good. Next. Very good. After inferior vena cava, right here, right? This is now my inferior vena cava, right? So we started here, right here, systemic capillaries. So this one, the systemic capillaries of my legs, and then this was my popliteal, the saphenous, popliteal, femoral, and then now we landed in the inferior vena cava. Look at this picture. So this is still big. You saw this in your cat, right? So still big. No chance for clot to block anywhere. Then the next, right atrium, right, inferior vena cava, then right atrium, right? Still big. The heart is big. You saw your heart. Then your right AV valve, right ventricle. From the right ventricle, it will pump. This is called pulmonary artery. Then after pulmonary artery, pulmonary capillaries. And the capillary is the microscopic. So of all these veins there was my clot, it started here, thrombosis. Where is the first chance for that clot to cause a plug? Right here, in the capillaries of the lungs. And so that's why anywhere you have a clot, every time you see the word clot, you know vein. Clots only happen in veins, not in arteries. Only happens in veins. And so the first part that it will cause a blockage is always in capillary, not arteries, by the way, because arteries constrict. It's impossible to find clots in arteries unless you have an aneurysm. Okay? So if you have an aneurysm, which is common among smokers, but aneurysm can also just be present in anyone. They can, especially the cerebral aneurysm, that one is idiopathic. So there's no predisposing cause or factor for that, but smokers usually will have that aneurysm too. Okay? So because they're able to contract, not common to have aneurysm, not common to have clots here, common to have clots here. See, just by anatomy, you can explain now. You understand when a patient has deep venous thrombosis, you will see that DVT. You always now have to explain to your patients, be physically active and be conscientious. Be faithful in taking your blood thinners. Why? Because you can have a massive embolism that will block your arteries, your capillaries in your lungs, which is the key for gas exchange. That's how you get rid of your carbon dioxide, and that's how you pick up your oxygen. So you can die. You understand? I hope you remember this. I remember in the final exam, the comprehensive final exam, in the past, there was a question. I said, write, and not I said, but in the test bank, it says write in detail or write down the blood vessels involved in the flow of blood from here. And it's not that hard. You just have to remember saphenous, then popliteal, then femoral, then inferior vena cava, and then from there, you remember this one already. The reason why I love that question in the test is because when you start working, you're going to encounter this one, thrombosis, very, very common. So always remember this flow chart. It is so scary, but it's a common problem. Maybe you can even take pictures of this, and this is your take home picture to your patients. You show them. You say the capillary is the one that is so small. That's the first chance for blood clot to form, to plug, and you show them. So this is big, 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 big. This is the first for that clot, and this is where you have gas exchange, and that will maybe, hopefully, encourage them to take their blood thinners all the time. You have any questions? Your test will also ask about different blood flows. So this one, um, remember, so this portal circulation, the blood flow to your lungs. This one, remember, the pulmonary artery carries deoxygenated blood. The pulmonary vein carries oxygenated blood. You remember the collateral circulation, how the coronary arteries have a right and a left to help, right, to make sure that even though the right coronary artery is plugged, you still have blood supply to your heart. Another very important organ that has collateral circulation, your brain. You remember the circle of Willis? Remember that? Here, your internal carotids here, and then here, your basilar arteries from your vertebrae. They meet at the base, circle of Willis, to make sure that your brain is properly supplied with blood. So heart and brain have collateral circulation. This one, portal circulation. If you're going to be asked for this one, you know. This should not be hard. These three, these two. And then femoral, here, femoral. Your saphenous, popliteal, your patella, right here, the kneecap. And then here is easy now. You go I think we forgot the iliac vein, but you forgot your iliac vein before vena cava. So femoral, then iliac vein, then vena cava. And that's it. Memorize that even for your final exam. Okay? You have any questions? Be sure you can read EKG. You will be given the choices. So you have the EKG and you're given the choices. Please pick one. Is this AFib, VFib, normal, tachycardia? Okay? So you'll be given the choices. And then you should be able to compute for a cardiac output equals heart rate times stroke volume. If you know the formula, if I ask you if you're given cardiac output and stroke volume, you can also compute for the heart rate, right? Or if you're given the cardiac output and heart rate, you can compute for stroke volume because you know the formula. You need to compute for net filtration pressure, and then your inotropic, chronotropic, your cardiac, your preload, afterload cardiac contractility, and then your cardiac cycle picture. Okay? So there's plenty of extra credit for Wednesday's test. Do you have any questions? Let's have a break for 5 minutes, then we're going to study our next body system, our lymphatic system.